So I'm going to start things off with a little bit of a general introduction, and then I'm going to turn it over to Dr. Reed to introduce Dr. Barr. And Dr. Barr, I'll just mention that it looks like people are going to be joining as we figure out the right way to get here. So I apologize for that. But welcome everyone to the Huck Distinguished Lecture Series. My name is Troy Ott. I'm a professor in animal science and associate director of the Huck. I'd like to take a moment to thank our faculty lead for the Huck Distinguished Lecture Series, Dr. Laura Wayrich, who is actually teaching class right now, and also Melissa Bailey and other members of the Huck staff for their assistance in organizing these exciting lectures. Today, Dr. Barr will be introduced by Andrew Reed, director of the Huck Institutes. And following the talk, Andrew and I will moderate a Q&A session with Dr. Barr. In this Q&A session, members of the audience will be able to ask their questions either in the chat function or by raising your blue hand. And Dr. Reed will be looking for the blue hands, and I'll be reading the chats. And so between the two of us, we'll try to field these calls. Also, as soon as I get started here, I'm going to hit the record button, and this lecture will be made publicly available on the Huck YouTube channel following the lecture. At this time, I'd like to ask each of you to ensure your microphones are muted, and I encourage each of you to turn on your videos if possible. Andrew, please introduce Dr. Barr as soon as I hit the record button. Hold on a second here. Laura, can you hit the record button? Laura, can you hit the record button?
I'm seeing a record button. Looks like it's going to me.
Good, great. We're great. All right, go ahead, Andrew.
Okay, thanks very much, Troy. Eliav, sorry about that disruptions there. It is my very great pleasure today to introduce you. Eliav Barr is the Senior Vice President and Head of Global Medical Affairs at Merck. His career began here at Penn State, and then he went to medical school at Jefferson, was a cardiologist resident at Hopkins, postdoc at Michigan, and was on the faculty at University of Chicago before moving to Merck. I first crossed paths with him when we were both independently visiting Princeton many years ago. As he was leaving our meeting, where actually he mostly asked me about my work, and my recollections, we mostly talked about mine. One of the faculty whispered to me after he left the room that this was the guy that was going to eradicate cervical cancer. And ever since then, I have to say, I've been very starstruck. I think the HPV cancer story is one of the best things that has happened in medicine and public health in my lifetime. And just, we went from sort of figuring out the cause to really being on the path to elimination in just a few decades. And Eliav has played a hugely outsized role in that incredible story. But he's not just an HPV guy. He's been a key player in Merck's very impressive and impactful efforts in HIV, in hepatitis C, and in COVID-19 therapeutics. And he actually offered to talk about any of those things as well. But I'm really, I was such a fan of the HPV story. I was very keen to hear his story. He was honored as Eberly College Outstanding Science Alumni in 2012, and in 2018, he was awarded the Penn State Alumni Fellow recipient. I have to say quite rightly, if you start to think about Penn Staters who have saved lives and stopped human misery, Eliav has to be one of the ones who's really up there numerically, and that's going to be a benefit to humanity for generations to go. Cervical cancer was a total fear for my mother's generation. And for my children's generation, it's going to be maybe just something banished to memory. So really quite something. Eliav, I'm really glad that you agreed to come and give this talk. I'm very much looking forward to what you have to say. Thanks very much.
Well, thank you very, very much for the opportunity and for the kind words. I really, really appreciate it. It's always a joy to talk about the story of HPV vaccine. And what I'm going to do is I'll share my screen, and hopefully you will let me know if you see what you see the slides. Do you see it?
Mm-hmm.
Good. Let me just make sure I got it duplicate. Right. Okay. I hope everyone can see the slides. You know, I just want to remind folks that I am an employee of Merck, and it's the maker of the 4 9-valent HPV vaccines, therefore that's my disclosure. Okay, So what I wanted to talk about was the arc of the development program for HPV, starting with the fact that HPV causes cervical cancer, but it also causes a lot of other cancers, as well as anogenital warts and recurrent respiratory papillomatosis cases. The discovery of HPV as the cause of these cancers led directly to a vaccine. And in trials of about 50,000 girls and women and boys or men, we showed that the vaccine, when given in a prophylactic, that is prior to infection setting, was highly efficacious. Now we have real world effectiveness trials that have confirmed phase 3 results, and I'll show you some of that. And then finally, we have to understand that clinical trials are just the beginning of the process, because one has to ensure that the intervention is used, that people have access to it, and that we can create vaccination programs that will ultimately result in eradication of HPV around the world. So let's start with the basics about the HPV and the development of the 9-valent vaccine. So the virus itself is a very common virus. It's a double-stranded, non-enveloped DNA virus, 8 kilobases. There are about 100 different types of HPV, but a certain proportion of them infect humans, and a certain proportion of those cause disease. HPV is the most common sexually transmitted infection in the US. Approximately 43 million Americans have HPV infection. Individuals acquire HPV infection during sexual intercourse, including oral sex, and the highest incident rates are in adolescents and young adults. The risk increases with a number of unique sexual partners, and most HPV infections resolve, but chronic infection leads to a set of events that ultimately can result in cancer. Now, this is a schema of what's called the transformation zone, and the transformation zone is a part of the body where wet mucosa and keratinized mucosa meet. So it's kind of every orifice that's one has ultimately leads to a border between squamous epithelium and columnar epithelium that compete with each other in an area called the transformation zone, and that's the favorite place for HPV to infect. And as you can see with that middle sort of orangish stuff here, the virus infects the basal epithelium, and as the cells mature, as they get closer and closer to the surface, in normal terms, they kind of mature and then senesce and become nothing more than keratinized kind of protein packets. Now, HPV infects the basal epithelium, and one of the HPV, or two of the HPV proteins, E6 and E7, results in a transformation of the cell into continuing replication. Initially, that replication does not prevent the cell from maturing, and during the maturation step, when keratin production is turned on, the outer coat of the protein of the virus, the L1 protein and the L2 proteins are expressed, and so the virus can be a full virus particle can be made. When the keratinized cells slough off, they contain a whole bunch of infectious virus. Ultimately, overexpression of E6 and E7 leads to a second hit of some sort, unknown at present, which leads to nonstop proliferation. And that proliferation actually ends the ability to produce productive virus, but it does cause oncogenesis and ultimately invasive cancer. And again, these are all, these can be in the mucosa of the anal canal, it can be in the mucosa of the cervix, it can be in the mucosa of the throat. And each of these are locations where cancer can occur. Trying to treat HPV has different kinds of, there's different kinds of approaches, of course. If you're already infected, there's always surgery to remove the lesion, but there's high rates of recurrence, and this is obviously quite disfiguring, both in terms of things like excising precancerous lesions using surgery to remove part of the cervix, or in the case of cancer, to have a more radical kind of approaches. There have been attempts to create vaccines that inhibit the functions of HPV, but these have not really worked very well. Same is true with small molecule inhibitors. Prophylactic vaccines seem to have done to be the right place to intervene, because you can prevent the infection from the get-go and prevent this process from occurring. But because HPV is so very common, and vaccines are only relatively new, there's a lot of information about what HPV causes around the world. And of course, as Andrew mentioned, cervical cancer is by far the most common of these cancers. We have about 530,000 new cases around the world, 275,000 deaths. And most of these cases can be prevented by cervical screening, by Pap testing. About 75% of them can be prevented, but that again requires nonstop screening, but also a lot of surgery, which ultimately is not particularly welcomed by women and can lead to fertility issues. Other cancers are also caused by HPV. 90% of anal cancers, 25% of vulvar cancers, 70% of vaginal cancers, and genital warts as well. Genital warts, I would say, they're not cancerous, but they cause a lot of trauma, both in terms of the treatment, which is excisional and repeated excisions, and a whole lot of heartache among young adults. Oropharyngeal cancers, particularly the cancer of the tonsil and the base of the mouth, are caused by HPV. The number of cases is about 22,000, but it's probably underestimated because of the fact that many of throat cancers are not really tested for HPV, except in the United States and in other wealthy countries where the treatment course can be modified by whether the tumor is HPV positive or not. RRP is a very rare event, but a very devastating event. It's caused by the HPV types that also cause genital warts, and essentially is a benign growth that is very, very aggressive and causes severe problems in babies because they have repeated need for surgery to avoid respiratory impairment. Now, as I mentioned, there are over 100 HPV types, but 9 of them cause most of the disease. HPV 6 and 11, among the life-threatening diseases, HPV 6 and 11 only causes recurrent respiratory papillomatosis. On the other hand, HPV 16 and 18 are the 2 most common HPV types that cause cancer. HPV 16 being absolutely the most important one. To get by itself, it's responsible for half of all cervical cancers and almost all of the remaining ones. But when you add 16, 18, and then another 5, you can get coverage to over 90%, to 90, 91% of cervical cancers, and you can see a large majority of HPV-related cancers elsewhere. As I mentioned, genital warts and precancerous lesions can be caused by a variety of cancers, and genital warts are 90% caused by 6 and 11, and by removing them, one can greatly reduce trauma to young adults. And from a health economics point of view, because the vaccine is given to adolescents, and because the genital warts happen very soon after sexual debut, one can already start to see tremendous savings in terms of prevention of these particular lesions, and in so doing can magnify the cost effectiveness of vaccination. Other precancerous lesions that are the subject of repeat follow-up through Pap testing or surgery are overwhelmingly caused by these 9 types as well. So we have 9 HPV types that cause a tremendous amount of disease and then we can figure out a vaccine that essentially is the outer coat protein of the virus manufactured in yeast cells. And we started to do studies of this vaccine in 1997, and the research program has continued since then, but we've already had clinical trials in a very large number of patients around the world, or subjects, I should say, around the world. So the original vaccine was 4 types, 6 and 11, which represents 90% of genital warts, and 16 and 18, which together accounted for 70% of cancers. The patient population or the subject population that we evaluated were 16 to 26-year-old women and 16 to 26-year-old men, primarily because sexual debut occurs in most parts of the world in the mid-teens. What we were able to demonstrate that when we administer the vaccine, the reduction for development of new cases of disease was quite striking. We see for low-grade cervical intraepithelial neoplasia, that is the precancerous lesions, 96 to 98%, whether it's low-grade or high-grade. And the same numbers are really quite striking across the board. Efficacy in men is slightly lower, and there's a lot of hypotheses around this. Perhaps it's having to do with the fact that the target organs are heavily keratinized. And perhaps maybe there's some limitation to the antibodies that are formed by the vaccine. Nevertheless, with respect to high-grade anal cancer and to genital warts, efficacy is better than most vaccines, with a low of 75, but in general 89 to 100. The Ninevalent vaccine was the second generation and really is designed to reduce the number of cancer-causing types so dramatically that it might have an impact on the need and the cadence of screening. So we developed this vaccine, about 4 or 5 years after the first vaccine was licensed. And what we did was we replicated the immunogenicity and the kind of antigen load of the quadrivalent vaccine a little bit more, simply because we wanted to avoid interference, immune interference. And then we added the types, the 31, 33, 45, 52, and 58, which together accounted for about 20% of cervical cancers, in order to increase the reach of the vaccine. There we demonstrated efficacy. So, what we were able to see was that in terms of the types in the 9-valent vaccine compared to the 4-valent vaccine, when we look at the old types, the old HPV types that were present in both of the vaccines, the rate of disease was comparable. In fact, maybe a little bit lower than we saw in many of these cases with the old vaccine. And that points out that we didn't lose the protection for the 4 types that were responsible for the majority of cervical cancer cases. And then when we looked at the new types, we were able to show again the same kind of super high efficacy against both infection, cervical disease, and vulvar and vaginal disease. So in women, the efficacy of the vaccine, the new vaccine that included 9 types, was comparable for the original types in the old vaccine, plus the 5 new types. Together, therefore, an overall efficacy that probably compared to placebo, we could impute to be in the upper 90s. And enables us to cover 90% of all HPV-related cancers and 90% of all genital warts. Vaccine manufacturers take on the responsibility to look at the long-term effectiveness of the vaccine, because it's really important to know whether you need to have booster. In order to make sure that you can continue the positive effect that one saw in the clinical trials. For HPV, the risk of acquisition is lifelong. As long as people are sexually active, they have a risk of acquiring HPV. And so it was necessary to look to see whether the vaccine could last for long periods of time. One way that we can do that is through registries and integrated healthcare systems that enable us to follow people post-vaccination over the duration of their lives. And we can do that in Scandinavia, which has a tremendous registry system that's really quite good in following individual patients and ensuring that they get screened for HPV. And so what we looked to see was whether vaccinated women who were enrolled in Scandinavia over the years, did they ever develop any more HPV 16 and 18 related disease in their cervix. And as you can see, by the happy number of 0, there is no new cases in these patients now 4, 6, 8, 10, 12, 14. We've continued to do this now, and the numbers have increased as women got older. And again, we see no waning of the effect. So with that, we ended up with the FDA approved indication for the 9-valent vaccine and similar ones around the world. This is a whole list of things that it caused that is indicated for. But the key thing here is that the vaccine can be administered from girls as young as 9 through 45 years old. The optimal age is really before sexual debut. And this is for this reason in the United States and many parts of the world, vaccine is administered around the age of 12 and 13. The diseases that the vaccine is meant to prevent are all of the cervical, vulvar, vaginal, anal, oropharyngeal, and other head and neck cancers that are caused by HPV types in the vaccine, as well as genital warts, but also all the precancerous lesions that are associated with these types. And this particular element is really important because, again, as governments consider whether this vaccine is relevant and affordable in their countries, by showing that you can eliminate all of the things that happen to women as they get pap tested over the course of their lifetime, if you can show that they didn't need to undergo further testing, that didn't need to undergo excision, they didn't need to have the resource use and as well as the psychological impact, then you can show that the vaccine is highly cost effective. And in the same manner with boys, although boys don't get screened for cancer unless they are gay men in certain parts of the world. The regimen that we use is a 2-dose regimen or a 3-dose regimen, and the schedule is for 2 doses is 0-6 or 0-12 months. The 2-dose regimen in younger kids it creates the same immune response as a 3-dose regimen in the population for which efficacy was shown. And this also points out some of the efficiency of vaccinating younger folks because their immune response is so much better than even young adults that all you need is 2 doses. And you can imagine that it's much easier and much less expensive to use 2 doses of vaccine and get full protection compared to 3 doses if you were to vaccinate somewhat older people. Now, it's important to remember that the vaccine doesn't prevent infection or doesn't prevent cancer in people already infected, and therefore there's a need for women to undergo cervical cancer screening, depending on when people finish or start and finish their vaccines. It could be that they were already been infected with HPV. It's also fair to say that the coverage of HPV in the United States is okay, not great. It's improving, but obviously that means that cervical cancer screening needs to continue. It's important to remember that that the vaccine is not a treatment for lesions that are already present, and not every precancerous lesion is prevented. And the reason for that is that there's rare cases of cancers caused by types that are not in the vaccine. There's also safety issues that we have to consider, and that's always important when you think about vaccination programs. Some people over the course of their young lives will have hypersensitivity to vaccine components, and some of those can be impactful with HPV vaccine. There was a very interesting phenomenon early on after the vaccine was implemented, that young girls developed syncope, and the syncope oftentimes happened before the vaccination or at the time of needle stick, and so there's a lot of there was a lot of mass events that happened like that, but with careful approach both to the subject and their parents and how you give the vaccine in terms of calming people down, that syncope went away. The vaccine has not been, the safety and effectiveness of the vaccine has not formally in clinical trials been established in pregnant women, although there's a large experience already in that group. And although we followed people for 14, 15 years, we haven't followed them for their entire lifetime. And so we don't know what the duration of the immunity that's engendered by the vaccine will last. I can tell you that it lasts a good 15 years, but after that, we have to wait to see as the studies continue to mature. Another thing that's important to remember about all intramuscular injections is that there's injection site adverse experiences, and the pain, swelling, and erythema are quite common. A little bit of headache, on occasion, a little bit of a temperature for people, especially after the last dose of the vaccine. Typically, these sorts of adverse experiences, both the local and the systemic, are caused by the immune response that's engendered by introduction of the antigens. And in a certain sense, it's kind of a no pain, no gain scenario. So the development program, and I'll talk now about implementation after this, showed that the vaccine is extraordinarily effective in preventing the infections that cause cancer and all of the lesions that lead up to the cancer. We didn't show in the clinical program that the vaccines prevented cancer. That happened later. But all the lesions, all the way up to carcinoma in situ, we could show prevention. Efficacy is at least up to 14 years of follow-up, so a total of 15 years. And then the vaccine should substantially reduce the risk of all these cancers. And we couldn't tell at the end of the clinical trial program whether this was true, but then subsequently this was demonstrated. So now let's talk about how the vaccine was implemented in the real world. And it's important to do that because, clinical trials, as I mentioned, are just that. They're tests. What really makes for a public health intervention is its actual use in the real world. And the expectation is that the behavior will be just like in clinical trials, but one always has to measure that. And what we were able to show, happily, was that we really did have a substantial impact on HPV-related disease, both in terms of actual lesions and cancers, but also costs and quality of life benefits. And, I used to have, until last year, slides showing sort of real-world effectiveness demonstrations with precancerous lesions. But the exciting bit of news that was published in the New England Journal last year now shows reductions in cancer. And the way that this was evaluated was vaccination programs in Sweden. As I mentioned, Sweden is one of the countries that has a tremendous public health system that enables individual patients to be followed through their lives using electronic health records, including vaccination, screening, and ultimately diagnosis of disease. Furthermore, in Sweden, there's a very active HPV screening program, and the results of those are also put into the EHR. So Sweden approved the HPV vaccine the moment it was available. It was licensed, and then it's used. They've used the quadrivalent HPV vaccine that we developed almost exclusively. Now they use a 9-valent vaccine. Adolescent girls were the first to offer to get the vaccine, and so that cohort of girls who initially got the vaccine were followed for the duration of their lives, and as they continue now, and compared to cohorts that did not get the vaccine. And so essentially this enabled the investigators in Sweden to look at the real world effectiveness of the vaccine in comparing women who did it or who did not get the vaccination. And they used 2 strata. The most important one was those vaccinated before age 17, which was heavily enriching for those before sexual debut, that is free of HPV vaccination at baseline, versus those who were a more mixed bag of those who had already been infected and those who were still uninfected. Remember that the vaccine is not effective in clearing infection. It's effective in preventing incident infection. So whereas I was really proud of a trial program of 50,000 people, there's nothing like real world effectiveness to humble a clinical trialist like myself. There was a total in this particular evaluation, there were 1,667,983 girls and women who were identified, of which 527,000 were vaccinated, and those were compared to the remainder who were not vaccinated. There were statistical adjustments made to account for baseline factors that may influence risk of HPV infection. And the key number is the incident rate ratio, which is essentially the rate of cervical cancer in vaccinated versus non-vaccinated individuals. The figure in the green below is really what I think is important, because this is the population that's ideally suited for HPV vaccination, now being followed up for a period of about 15 years, but with sufficient numbers to actually show cervical cancer rates. And as you can see, that the crude incidence was a drop of 90% relative in cervical cancer relative to the unvaccinated, so a incident rate ratio of 0.1. And after various adjustments, the overall period with age only and then with all the other factors really brought us back to an 88% reduction. So in the real world, vaccination actually did prevent cervical cancer from occurring. And those girls who had been vaccinated early on, and mostly prior to sexual debut, had a 88 to 90% reduction in their risk of cervical cancer. Genital warts is also important. And here we used Australia to determine what it means, what the vaccination program meant with respect to genital warts, because from a governmental point of view, if you could prevent genital warts, the cost effectiveness of the vaccine goes up dramatically because of a reduction in resource utilization. Genital warts are incredibly common, and we used Australia because Australia, like the UK, has a particular specialty called genitourinary medicine, that is folks dealing with STDs. And the registries in 2 states, New South Wales and Victoria, were able to be used to determine how the rates of genital warts progressed over the years. And we could look at it by strata that included those who are likely vaccinated versus not. So in 2007, Australia introduced the HPV vaccine, and uptake in both Victoria and New South Wales was quite vigorous. The initial vaccination was in young adults, and was in children. Adolescents with a catch-up population up to age 17, and then there was afterwards expansion to include young adults as well. So if you look at the blue line, that's the kind of the middle line. That's the vaccine. That's the population that received the highest amounts of vaccine. And you can see over the course of the years in that population, the number of people who had a new genital warts diagnosis approached 0%. And eventually the 21 to 30-year-olds drifted down as well as catch-up vaccination occurred. On the other hand, those who are over 30 years old during the vaccination campaigns, their rates remained about the same. So it basically taught us that genital warts were also dramatically reduced in countries that had high uptake of the vaccine and had the ability to measure the presence of this disease. So, now we can say that, HPV causes cancer. A vaccine against HPV can prevent infections and the precancerous lesions, and a vaccine against HPV can also prevent those cancers. And that represents a pretty strong story. But we also need to think about HPV elimination. And here we need to leave the realm of pure science and go into more social policy kind of questions. And there's a lot of questions about how do you administer a vaccine to a population of adolescents. It can be expensive. Adolescents are notoriously difficult to get to do anything. There's a lot of vaccine hesitancy. What's the best way to eliminate HPV? Do you just vaccinate the girls, or do you vaccinate both girls and boys? Well, a lot of work was done to model all of this, but also there's a lot of questions about things like ethics and things like why should girls bear the burden of HPV elimination when boys are also responsible and also infected. So the bottom line is that gender-neutral vaccination is really the most equitable, resilient, and effective means to control HPV for the reasons listed here. The basis for that I can explain in subsequent trials. Achieving equity is really important, obviously, because we want to make sure that both girls and boys are protected. Males have the opportunity to reduce their personal risk of HPV disease. While it's true that if we can get up to 100% coverage or very high levels of coverage in women, heterosexual men are unlikely to get infected, but not many countries have reached that level of coverage. Female-only vaccination policy creates a dilemma in which women are asked to assume all responsibility for vaccination, and they certainly don't. The vaccination programs don't help men who have sex with men or those who move outside the herd. And then the other problem is that when there are situations where women might not be accessible, having vaccinated men helps in the resilience of the program that is maintaining all of the strong protection that vaccination programs have achieved. And a good example of why female vaccination doesn't help everyone is that MSMs have high rates of HPV infection, and we can see that men who have sex with men don't get benefit from female-only vaccination programs. High-risk HPV infection is more common in MSM, and anal HPV infection, of course, is more common in men in MSM versus men having sex with women. So what we can see is that in Australia, where there was a girl-only program that included catch-up, heterosexual men had a nice decline in their in the rates of infection, but certainly in here genital warts over the course of the few years after the initiation of the program, but gay men did not. And in fact, these data, together with the results that showed that the vaccine prevented anal precancers, led Australia to implement gender-neutral vaccination. So we don't have any further data looking at heterosexual versus MSM. So as we stand today, the biggest impediment to and forgetting the COVID crisis for the time being, the biggest issue that we've dealt with so far is hesitancy in the part of the population, getting enough supply to vaccinate the world, although the company is now committed a couple of $2 billion, and we've spent that money to build new factories, and the logistical difficulties of two-dose vaccination strategies in kids or in adolescents. Still, I think the story is really good that we have a vaccine that is tremendously effective, a clinical trials set that has shown that efficacy under the best controlled opportunities, a real-world effectiveness data set that's unparalleled, that have shown the phase 3 results and show cervical cancer rates being reduced, and a lot of thinking about what's the optimal way to implement the vaccine, gender-neutral vaccination being the most important one. I would also point out that Merck is, we haven't stopped our research. One of the things that we've chosen to do, because the Bill and Melinda Gates Foundation, governments, and other NGOs have told us that two-dose vaccination is really hard to implement in low resource settings. So we've now started to develop a single-dose HPV vaccine with a new adjuvant that would induce immune responses that are sufficiently robust, that hopefully will have the kinds of protective levels that are seen elsewhere, that are seen with a two-dose vaccine, but that will require clinical trials. The prototype vaccine is in phase 1 development. But if you can get a single-dose HPV vaccine, that would make things even easier, and hopefully we'll be able to achieve universal HPV eradication. So with that, I'll say thank you very much for your attention. I'm glad to answer any questions, and hopefully you found this informative.
Thanks very much, Eliav. That was hugely interesting, and what a great story. It's fantastic. It works so well. We'll take questions either through the chat or with hands up, but I'm going to take the privilege of asking the first one, if I may. You mentioned that the company takes responsibility for following what goes on in terms of the waning immunity or not. Also, is anybody looking at the strain variation? Is there changes in strain frequency, any sign of serotype replacement, or any onward evolution of the virus?
Great question, and the good news is that there isn't any replacement, and there's not any evolution due to pressure. And biologically this makes sense, because this is a double-stranded DNA virus with good editing capabilities, because it uses the host DNA polymerase, and so it's actually a pretty robust and stable robust editing function, so a stable virus. We look very carefully at compartment effects, worried that if the compartment is empty of HPV-16, you'll find some other virus that crops up. Hasn't happened. Because data didn't show you, is we always had background rates of the HPV types that were not in the vaccine to make sure that those rates were comparable. And the same is true in Scandinavia, where they've looked back at biopsy specimens, and they're not finding any increase in those viruses that are not in the vaccine.
Oh, that's terrific news. Long may that continue too. Also wondering what you think is the major problems with getting this rolled out at high levels in the US. Obviously, it's possible in Australia and Sweden.
Yeah, I think that the U.S. healthcare system always has its strengths and weaknesses. The problem that I see is that they don't have enough. There's not enough political will to do it. There's enough vaccine, but school mandates are not enforced in many states. Part of the reason is because there's a different perception of viruses that are transmitted sexually versus viruses that are transmitted through respiratory tract, and governments have and jurisdictions, local jurisdictions might have different ideas about that. Vaccine hesitancy for HPV vaccine seems to have been we seem to have been a great lightning rod for vaccine hesitancy and anti-vaccine effect activism. And so that's been a bit of a thing. But over the course of the years, we've gotten better and better. So now coverage is at the 65% range, maybe 70 in some states. This is pre-COVID. I have to say that during COVID, we took a pretty significant hit. But hopefully as the pandemic ends, we'll be able to continue this for this slow upward movement and continue persuading parents and kids to get the vaccine.
I have a question in the chat box, if I may, Andrew. So the question is, in the current vaccine schedules, are young women who only got the original vaccine recommended to get the newer one in addition?
It's up to them. There's not a. It's not a must recommendation. You can get a boost of the with the 9-valent vaccine. It will be efficacious for the 5 types, and we've done studies like that. But it's not in the indications, and therefore not in the vaccination schedule.
Any other questions?
I have a question, and this is an area that's slightly outside of my expertise, but does the fact that vaccination after exposure to the virus, does the fact that vaccination doesn't reduce development of disease suggest anything about the progression of carcinogenesis? You talked about there being a 2-hit model, and one would think if you were at least able to suppress the amount of virus around or the amount of virus replication, that you should reduce the incidence of disease, even if you didn't eliminate them completely.
It's a terrific question, one that hasn't that's difficult to answer. One theory is that the antibody levels are so robust that you never get any actual infectivity in the epithelial layers. I don't believe that, because the levels wane over some significant period of time. Others say yes, the levels wane over a significant period of time, but you constantly get exposed whenever you have sex, and so you actually have mini boosts all the time. And so it's not clear. What I think is I'm not aware of any studies that have tried to measure viral load and say, well, vaccinated people have less of a viral load. So that's unfortunately an issue that's very difficult to assess. What we do know is that the natural history of the disease among people who are infected with a particular type of HPV at baseline, and then they get the vaccine, is not impacted, not in a positive or negative way. So, there's no evidence to suggest any beneficial effects or any effects that might occur if virus load is reduced.
Thank you.
Eliav, I wonder if you could say a few words too about Merck's decision to take this on, because presumably it wasn't immediately clear that this was going to work as well as it has. If I remember correctly, there was only one other pharma company that got into this game.
That's right.
So what made you guys jump for it and almost nobody else?
Well, we started off, well, first of all, we had experience before with hepatitis B vaccine. There was also a virus-like particle, and it was also a cancer-causing virus. And so we thought, well, it's a different virus, but we certainly have the technology to make that particular VLP. Then the next piece after that was the fact that this was a huge public health need. And, it's rare that you can try to do something that's cancer prevention. That was really, very, very enticing in that regard. And, our philosophy has always been, if there's a huge unmet medical need and a intervention that really works, then the profits will follow because it's a major public health benefit. And that the enthusiasm for participation in clinical trials, the ease of which people are persuaded to participate, is a good proxy for whether the society values the product. And so in thinking through all of that, and in working with epidemiologists who had actually done all the natural history studies, it became clear to us that if this worked, it would really make a big difference for women, and ultimately we found for men. And so that's what happened. It was a huge gamble. It was the clinical program, 50,000 people clinical program is a massive endeavor and hugely costly, but at the same time, the results have been quite spectacular, and it's turned out to be good. Not everything works. We just had 2 COVID vaccines that didn't meet the mark, and there are many others that have failed along the way in other parts. But that's science. It's not easy.
And dare I ask, in the Merck portfolio, is this one of your big hits, or is this is one of your big success? I don't mean in terms of the impact on health. I mean financially. Is this
financially? It's been a huge. It's been an important part of our portfolio, and the people think that, okay, you finish the clinical trials and you're done and now it's all profit, but actually it's not the case. We've been doing clinical studies for quite some time, and as I mentioned, investing in factories, upgrading processes, and so on. Right now we're just finished a lot of the vaccination program in China that needed to be repeated because China wants to do their own thing. It's a massive hit in China right now. There's a tremendous desire for the vaccine. So yeah, it's really quite been quite rewarding for us. But I think it's paid for a lot of stuff that didn't work. That is a story for another day.
Yeah, right. But I mean, it is really a fantastic story. Are there any final questions before we let Eliav go? Okay, in that case, I'd like to say thanks very much, Eliav, for a really stimulating talk, and as importantly, for the fantastic work that really is making a huge dent on what we hope will soon be a major health problem of the past. So thanks very much for the talk and very much for the work, Eliav.
Thanks for the invite, and have a great rest of the day.
You bet.
Yes, thank you.
Take care. Bye-bye.